So, in this video, we are explaining the Hardy-Weinberg model, which is basically the idea of looking at the genes in a population. And then we're going to talk about how you mess up the Hardy-Weinberg model. Basically, what causes evolution, the general cause of evolution, and the timeline, how it actually happens. So, what is evolution, first off, is a change in population through time. And there are two types. There's microevolution and macroevolution. Microevolution is a change within species that occurs over dozens or hundreds of generations, so a small amount of time. And it is also known as population genetics. Macroevolution is when you talk about things that are in a much longer period of time. It includes making of a new species. So microevolution is when one species changes over time in a short amount of time, and macroevolution is when one species becomes either a brand new species or two new species or three new species in a much longer period of time. And that's generally what people think of with evolution is the macro idea. What we're going to focus on is the microevolution or population genetics. What actually causes evolution is you have to first start off with variation in your population. And what causes variation is genetics. It's all about the genes that various people have and how they mix and match together. And that's what allows populations to adapt and evolve to new environmental conditions. And that is the basic raw material for evolution and is absolutely necessary in order for a population to evolve. So, we have to first understand what a gene pool is. Gene pool is all of the genes in the local population of an organism. So a gene pool of a biology class would be all of the genes of the people in that class. So every single one of them. And it's one local population, not the entire species, which is important because you have to realize it has to be a local population because those are the organisms that are going to interbreed. The outside organisms, even though if they're in the same species, if they're on a different continent, they're not going to be part of that gene pool because their genes are never going to get into that one population. So, what the Hardy-Weinberg model states is a mathematical model of gene pools and how they relate. So these two guys, Godfrey Hardy and Wilhelm Weinberg, recognized that there is a simple mathematical relationship in the allele frequencies in gene pools. That then can predict the genotypic frequency in the next generation by saying, okay, this gene pool has this allele frequency, and therefore the next generation should have this genotypic frequency, which makes tons of sense, of course. It's not confusing whatsoever. But to put it into an easier way of looking at it, we have to go back to genetics and Punnett squares. So you have the allele frequency. So say you have the allele frequency for red flower is P, and the allele frequency for white flower is Q. Then, therefore, P plus Q equals 1. Because remember, the alleles frequencies have to equal 1. If you only have two of them, then the addition of each one of them have to equal 1. So then when you look at the genotypic frequency, you take those, so P and Q, remember P is for red flowers, Q is for white flowers, to figure out the frequency of a plant that are homozygous for the allele that causes red flowers will have to be P squared because you have two of them. Then, in order to find the frequency of a plant that's homozygous for white flowers, it would be Q squared because, again, two of them, you have to have two Qs. You have to have two white alleles for a white flower to be homozygous. Homozygous for red, you have to have two red alleles, so therefore P times P is P squared, Q times 2 is Q squared, and then the frequency for the heterozygous plant would be 2PQ, because heterozygous would have 1P and 1Q, and there's two of them because, if you remember back to genetics, you had a 1 to 2 to 1 ratio. And so the expected genotypic frequencies for the next generation would be p squared plus 2pq plus q squared equals 1. So if you look at the actual Punnett square, you'll see here's our idea. If you have two heterozygous, you assume with the frequencies, and you would actually have frequencies in here, but this is where you'd have the P and Q and P and Q, and you assume heterozygous cross, and you'd end up with your P squared, as you see, your Q squared in those boxes, and then the 2PQ come from the fact that there would be two boxes with PQ, so hence the 2PQ. So basically, one allele frequency depends on the other and the past generations, and no allele can go away. Even if in one generation you don't see any white flowers, that doesn't mean that that allele went away. You just have more heterozygous, because you cannot ignore and lose an allele. They get passed on from one generation to the next. It's just simple science. Now, what actually happens is that all of them don't get passed on. The Hardy-Weinberg simplifies the situation and makes a few assumptions. First, there's no mutations, which is just silly. Second, nobody moves, so that gene pool stays constant, which is, again, silly. Population size is very large, which sometimes and often occurs, but there are some populations that are very small, and in a very small population, you might not have an allele, and you might actually lose an allele. And there's random mating. All the red flowers don't mate with the red flowers and white with white, but there is random, so that hence the heterozygous cross is assumed in random mating. And then lastly, that there's no natural selection. So how and when does this actually ever happen? All of these 5 things. So there's no mutation, no migration, large population, random mating, and natural selection doesn't occur. That's basically never. And this kind of proves that evolution occurs. So the Hardy-Weinberg model, they came up with this idea and then went, and this in and of itself, because this never occurs, proves that evolution does occur. So now we're going to look at messing up the Hardy-Weinberg equilibrium. We're going to look at each one of those assumptions and explain how they could actually cause evolution. So the first one, mutation. Spontaneous mutations always occur. They occur at a low rate. But we are often also exposed to mutagens, and so therefore the rate of mutations increases. Now, some are beneficial. Those are the really important ones for evolution. Some of them are not, but the bonus of the ones that are not beneficial, they don't get passed on to future generations, most likely because those people do not survive as well and cannot actually pass it on. So the beneficial ones get passed on more and then eventually become more common. So therefore the mutation becomes commonplace and you don't even realize it's a mutation anymore. All right. Now, this actually just shows through generations how good mutations can actually grow in a population. So we have population in the red is mutated. That is your mutated organism. Then in the next generation, that organism was able to have more babies. So it grew. And then in the next generation, it grew even more because those ones also had more babies and they were able to survive better. So they were able to have even more. The next generation, it might level off, but then eventually it becomes a commonplace mutation. It becomes more common than the unmutated. So therefore the one red that started in generation 1, now you have 5 reds and 1 blues. So that's how the good mutation grows. Now the second assumption is migration. That migration doesn't occur. Well, many organisms immigrate and emigrate from populations. If you look at the human population, we're moving all the time. And oftentimes this is behavioral for wild animals because they can't have more than 1 male in a herd. or they can't have too many males, or their food source dies out, so they go and find somewhere else, and they join with another group. So this migration just occurs all the time, and therefore the gene pool cannot stay constant, because if somebody leaves, they take their genes with them. So therefore your allele frequencies just changed. And this is known as gene flow. You have where your genes flow from one place to another. And this causes the allele frequencies to change in various populations, and therefore the Hardy-Weinberg model to not work. The third is population size. Genetic drift is a process that affects small populations, and it's when you have a change in allele frequency due to random events or chance. So suddenly killing off all the blondes. Then there would be no blondes and no blonde allele, or very few blonde alleles, because only the heterozygous would have that blonde allele. So it affects small populations more because there's just fewer of them. So it's kind of like if you had a class size of 10, if a whole bunch of you did really well on a test, but 2 of you did really poorly, that would bring down your entire class average a lot more than if there were 50 of you in the class and 2 of you did poorly. The class average wouldn't be affected as much. Same idea in small populations. If you have 10 dolphins and 2 of them die, well, that's a lot of your population, versus if you had 100 of them and only 2 of them died. That wouldn't be as much of a problem. So in small populations, alleles could even go away. And this just shows 2 pictures of the large versus small population. So you'll see on the left, that's a large population up top, and then it's going into small populations. And the frequencies, if you look on the far left, larger population has equal frequencies. The medium ground does not, and in the small population, you only have one type of gene. So you go from a very large population of a 50-50 for the frequencies to a very, very small, where it's 100% of one. And same thing with the picture on the right. You have up top the graph, the small populations allele frequencies, and you'll notice there are quite a few that go to 100 and down to 0, or 1 down to 0 for frequency. So one of the alleles took over, and the bottom one are larger populations, so you'll see majority of them are more consistent and more constant. They do change, obviously, but none of them really get lost except for one, you'll see, because that does occur. But as you see, affects large population. It affects large populations a lot less than small populations. Now, large populations can become inbred if there's what's known as a population bottleneck, which is a drastic reduction for a few generations, and that causes inbreeding, which inbreeding is problematic because it increases the frequency of the harmful and lethal homozygous alleles, and it's known as inbreeding depression, when their fertility and survival are reduced. So they have lots and lots of problems, because you just end up with a whole bunch of one type, which we've been discussing. Evolution needs variation. Inbreeding decreases variation, at a very simple term. And then there is the founder's effect, which is a version of large populations going to small populations. It's genetic drift that influences new populations. So a new population can have the allele frequency that's very different from its actual source population. So if you look on the right, you have an initial population of a lot of dark blues and one lighter. And those 4 that are circled go and form a new population. Well, in the initial population, that light blue wasn't that common, but because there are only 4 of them, in the new population it's 25% of it. And therefore, when it grows, you end up with a whole different population. An example of this is the Canada geese and the Hawaiian geese. So on the left picture, that's the Canadian geese, and then on the right you have the Hawaiian geese, and you'll notice all their differences. They came from the same population, but the group from Canada, a small group from Canada, flew to Hawaii and started their own population, and they just ended up being very different than the original population. And then the fourth assumption is non-random mating. Well, mating is usually due to geography, whoever is closest. And there's also assortative mating, which is mating with someone with similar traits. So it actually does occur. You mate with someone that you look like. And in the wild, it's really to make sure that you're mating with somebody that is one of your species. You don't want to mate with somebody that doesn't look like you, or else you'll be in big trouble if they're not your species. So the thing with the non-random mating and the fact that that doesn't occur doesn't affect the overall allele frequencies, but it does influence the phenotypic frequencies. Because if you have two brown-haired people mating, they'd produce most likely more brown-haired instead of blondes, and the blondes will produce more blondes. So that's the same idea. You're not killing off any alleles, you're not destroying the allele frequency or changing it, but phenotypic frequency, which is what evolution works on, what natural selection works on. Which brings us to the fifth one, natural selection. It is the most significant factor because the environment is always changing. Natural selection changes the frequency of a gene pool and helps the population to adapt. So there are many examples of natural selection, like the peppered moth, sickle cell anemia. All of those are types of natural selection. Again, they work on phenotypic frequencies because they affect your actual phenotype. And there are 4 different types of natural selection. Stabilizing selection, which is when the average is chosen for. So therefore, number of eggs in birds. So you don't want to just have lay one egg, but you don't want to lay too many eggs. So an average amount of eggs is a good idea, so that if one dies, it's not that big a deal, but you have enough food to actually keep those ones alive. Directional selection. That is when it goes from one extreme to the next. So for instance, a peppered moth went from the white peppered moth to a black peppered moth, so the extreme of white to the black. Then you have disruptive selection, which is when the 2 extremes are selected for. So black and white rabbits, the black rabbits fit better in the forest and the white rabbits fit better in the field. So there was no gray rabbit because they didn't fit anywhere. And so those actually usually turn into 2 different species eventually because they no longer interbreed. And then the last one is sexual selection, is the fact that you actually choose a mate that is the best mating partner, and therefore that trait will be chosen for. So with peacocks, the plume has to be big and colorful, and that is what is chosen for, so therefore the big plume and colorful plume will be passed on to future generations. So all of these are involved in natural selection and cause evolution to occur. And lastly, to look at examples of recent population evolution, we have sickle cell anemia. Sickle cell anemia is a genetic disorder that is usually problematic, but it also wards off malaria. So sickle cell anemia becomes huge in malaria-ridden countries because you don't get malaria. So you can survive with sickle cell anemia, you can't survive with malaria. So everyone that has sickle cell anemia doesn't get malaria and die, and then passes on that sickle cell anemia gene. Lactose tolerance. If you're lactose tolerant, you're actually a mutation. We became lactose tolerant as a species when we started domesticating cows, because before that we didn't have milk after birth. After you weaned off of your mother's milk, you never drank milk again. But when we started domesticating cows and were able to get milk from cows, we were able to start digesting it, and it was actually better to be able to break down and drink milk because it made you grow stronger and be able to actually have more children. So therefore that gene and that mutation got passed on more because it was a good mutation and allowed us to produce more babies. Pigmentation, all of our different pigments. And if you look at different places around the world, there are different colored skins. And it's due to having various sun amounts. So you want to have the lighter skin if you're not in a very sunny place, but you want to have darker skin if you're in the sun, because darker skin reflects the UV light, so you're less likely to get skin cancer. So all of these have actually been recently studied and have the 4 determining factors to show evolution. The determining factors are the now things that have to be shown in research to prove evolution, and it's the fact that it's a genetic, it's large scale, so it's not just one little population, it's basically worldwide or countrywide. It has to be geographically related, and it has to be statistically significant, obviously. So people are currently looking at humans mostly, and also some other organisms, but mostly humans, and looking at how we have evolved and where we are heading towards, and seeing what we can do with this. So it's all brand new, and even though evolution is looking at the past, it is also looking at the present, because we are evolving as we speak, and everybody always wants to know, well, what's next? So this will answer what's next.